This approach can help make the study of biochemistry more accessible and enjoyable for you.
So, for example, could you please design an outline for biochemistry, the foundation of biochemistry for a high school student?
You're asking for an outline of foundational biochemistry, suitable for high school students. Here's a basic outline. 1. Introduction to biochemistry, understanding the basic concepts and the importance of biochemistry in life sciences. 2. The structure and function of biomolecules, exploring proteins, carbohydrates, lipids, and nucleic acids. 3. Enzymes, learning about enzyme functions, types, and their role in metabolic pathways. 4. Cell structure and organelles, studying the different components of a cell and their functions. 5. Metabolism, an overview of metabolic pathways, including glycolysis, Krebs cycle, and oxidative phosphorylation. 6. DNA and RNA, understanding the structure and function of genetic material. 7. Protein synthesis, exploring how proteins are made from DNA blueprints. 8. Biochemical techniques, an introduction to common laboratory methods in biochemistry. Each topic would be presented in a way that's engaging and easy to understand, possibly using AI tools for interactive learning and real-world applications. This approach aims to build a strong foundation in biochemistry for high school students.
Okay, my next question is regarding senior research. For example, I'm a senior biochemist and researching on the biochemistry-related aspects, human health-related aspects. And can you design an advanced biochemistry courses, like AI-driven course, for me to study that?
Here's a suggested course outline. 1. Advanced molecular biology, delving into the complex interactions within cells and how these relate to human health. 2. Computational biochemistry, using AI and computational methods to analyze biochemical data. 3. Genomics and proteomics, exploring the genome and proteome and their implications for health and disease. 4. Metabolic disorders and biochemistry, studying the biochemical basis of diseases related to metabolism. 5. Pharmacology and drug design, understanding how drugs interact at the molecular level and how AI can aid in drug discovery. 6. Biotechnology and health, using biotechnological methods for health applications, including gene editing and therapy. 7. AI in diagnostic and therapeutic development, exploring how AI is revolutionizing diagnostics and treatment plans. 8. Research methods and ethics, covering advanced research techniques and ethical considerations in biochemistry research. Each section would involve deep dives into current research, utilizing AI tools for data analysis and modeling, tailored for an experienced biochemist. This course would aim to enhance your expertise and keep you abreast of the latest developments in AI applications in biochemistry.
Okay, could you please also design an outline for me to learn the advanced biochemistry?
Sure, go ahead with your next question or request. I'm here to help.
Okay, please generate the outline for advanced biochemistry, driven by AI.
Here's an academic poster designed for an advanced AI-driven biochemistry course. It visually represents the course topics and is tailored to appeal to senior biochemists and researchers. The design integrates elements of DNA structures, molecular imagery, AI influences, and relevant lab equipment, reflecting the blend of biochemistry and AI. You can check out the images in our conversation history.
Okay, can you read about the outline and the content in detail?
I'm sorry for any confusion, but I can't directly read or interpret visual content like images or text within images. However, the poster was designed to visually represent sections of an advanced AI-driven biochemistry course. These sections included 1. Advanced molecular biology, 2. Computational biochemistry, 3. Genomics and proteomics, 4. Metabolic disorders and biochemistry, 5. Pharmacology and drug design, 6. Biotechnology and health, 7. AI in diagnostic and therapeutic development, 8. Research methods and ethics. Each of these sections would cover in-depth topics relevant to a senior biochemist integrating AI applications in biochemistry research. The poster's visual elements, like DNA helices and molecular structures, are meant to symbolize these topics. If you have specific questions about any of these sections, feel free to ask.
Yes, I do have questions. That's regarding the self-study driven by AI facilitate the biochemistry study. So can the attenders or the like students, okay, yeah, whether it's a senior scientist or high school student, can ask you all kinds of questions for you to answer, and then you just adopt the mimic the student ask your question and continue ask the questions, and until you ask about 100, 200, or 300 questions and put in the database and analyze the level of the students, the question level students, and then analyze for the novel kind of questions and redundant questions and the basic questions, etc. Can you do that?
You're asking if attendees or students, whether they are senior scientists or high school students, can ask me various questions, and if I can adapt my responses to mimic a student asking questions, continuing to ask and analyze questions of different levels, including normal, redundant, and basic questions. Yes, I can answer questions from users with different levels of expertise, from high school students to senior scientists. I can adjust my responses to fit the complexity and nature of the questions asked. Whether the questions are about basic concepts, advanced topics, or even repetitive inquiries, I can provide tailored responses to each. Feel free to ask any question you have in mind, and I'll respond accordingly.
Okay, great. So that kind of questions, and based on the questions the student ask you, the attendees ask you, and you can analyze the level and how much basic knowledge the student already mastered before entering to the workshop, the AI-driven biochemistry course. Is that correct?
You're asking if, based on the questions students or attendees ask me, I can analyze their level of understanding and how much basic knowledge they have already mastered before entering the AI-driven biochemistry course. Yes, that's correct. By analyzing the questions asked, I can gauge a student's current level of understanding in biochemistry and related subjects. This helps in tailoring responses and educational materials to suit their knowledge level and learning needs. It's a useful way to assess what topics might need more focus or a different approach in teaching, especially in a course that combines complex fields like AI and biochemistry.
Great. Can I also import the whole file to you? For example, I would like the senior scientist to analyze the recent publication in Nature and Science or Cell, and you can analyze those papers for analysis and tell the learner and how much advance those people can advance in the field, and what is a major problem left there unsolved.
You're asking if I can analyze recent publications in journals like Nature, Science, or Cell, and then explain these analyses to learners, helping them understand the content and significance of these papers. Yes, I can assist in analyzing and summarizing scientific papers from prominent journals. While I can't access new content beyond my last training date in April 2023, I can provide insights into the methodologies, findings, and implications of research papers published up to that point. I can break down complex scientific concepts into more understandable terms, discuss the relevance of the research, and how it contributes to the field of biochemistry or related areas. This can be particularly useful for learners who are new to reading scientific literature or who are looking for a deeper understanding of specific studies.
Great. So finally, the question. So the AI-driven biochemistry study can also generate the topics for scientists or graduate students to study and to make their research direction. Is that correct?
You're asking if an AI-driven biochemistry study can also generate topics for scientists or graduate students to focus on in their research. Yes, that's correct. AI-driven tools can help identify emerging trends, gaps in the current research, and potential new avenues for exploration in biochemistry. This can be especially useful for graduate students and scientists who are deciding on a research direction. AI can analyze large volumes of scientific literature to suggest topics that are novel, have a high potential for impact, or are in line with current scientific advancements. This process can aid in making informed decisions about research topics, ensuring they are relevant and contribute meaningfully to the field.
Okay, great. Thank you. And my final question is regarding the daily practice. For example, in my workshop, I would like the student design the guide RNA for gene editing. And also, we would like to modify the skillful RNA structures, the three-dimensional structures, by make mutation of the nucleotides. And I would like to ask the student to write down the detailed procedure for their experiments. Can you go over their experiment procedure and make comments, make suggestions, and make detailed annotation and make notes to request them for specific attention to the experiments? Can you do that?
You're asking if I can review and comment on students' experiment procedures, specifically for designing guide RNA for gene editing and modifying the scale of RNA structures by mutating nucleotides, and if I can provide suggestions, detailed annotations, and notes for specific attention in their experiments. Yes, I can assist in this way. If a student provides a detailed description of their experimental procedure, I can review it and offer feedback. This would include suggesting improvements, identifying potential issues, and making annotations for specific steps that require careful attention. My feedback can help students refine their experimental designs, ensure accuracy, and potentially enhance the outcomes of their experiments. However, it's important to note that while I can provide guidance based on general scientific principles and practices, the feedback might not cover highly specialized or cutting-edge techniques that have emerged after my last training data.
Okay. So maybe additional question regarding the programming. So biochemistry related the algorithm and the programming issue. So for example, for drug discovery, for the protein structure, three-dimensional structure analysis, can you help with that during my workshop?
It seems like your question got cut off. Could you please provide more details or complete your question regarding programming in biochemistry, particularly in the context of drug discovery and protein studies?
Yes,
this will help me give you a more accurate and helpful response.
Yes, my question is whether you can guide the drug discovery and the protein structure analysis. If I import, for example, the GFP protein, can you modify it and generate what kind of a color it turned out? And also for GFP gene editing in bacteria, what kind of guide RNA, what kind of scaffold RNA can you enable it much more efficiently be edited?
You're asking if I can guide in drug discovery and protein structure analysis, specifically regarding modifying green fluorescent protein GFP to change its color, and about designing guide RNA for efficient GFP gene editing in bacteria. 1. GFP color modification. GFP color modification involves changing its amino acid sequence, which affects the chromophore, the light-emitting part of the protein. By altering specific amino acids near the chromophore, the emitted light's wavelength and thus the color can change. However, predicting the exact color change from a specific mutation requires complex computational modeling and understanding of protein structure. While I can provide general guidance on the process and principles, I don't have the capability to perform these specific computational predictions or modifications. 2. Guide RNA for GFP gene editing. Designing guide RNA for gene editing, like with CRISPR-Cas9 systems, involves selecting a sequence that targets the specific part of the GFP gene you want to edit. The efficiency of editing depends on the guide RNA's specificity and the accessibility of the target site in the genome. I can assist in understanding the principles behind designing guide RNA and provide general guidance on best practices for creating efficient guide RNA. However, actual design and prediction of efficiency require specialized bioinformatics tools and experimental validation. For both these tasks, detailed bioinformatics analysis and laboratory experiment are crucial. Advanced software and expertise in molecular biology are typically required for precise predictions and modifications. My role can be to provide foundational understanding and general guidance in these complex areas.
Great. So, in speaking of the protein structure studies and gene editing studies, can you help a student modify the Cas9 protein structure by mutation, in vitro mutation by a PCR strategy, and also help them to design the scaffold RNA structure by mutation, also in vitro, and then try to gain the highest efficiency in gene editing in terms of the Cas9 structure and also the RNA, guide RNA, and the scaffold RNA structure.
You're asking if I can help students with projects involving the modification of the Cas9 protein structure through in vitro mutations using PCR strategies, and also in designing scaffolds for RNA structures through mutations to achieve higher efficiency in gene editing. 1. Modifying Cas9 protein structure. To modify the Cas9 protein structure, students would need to understand which parts of the protein are crucial for its function and how mutations might affect its activity. This involves identifying specific amino acids for mutation, which can be informed by existing research on Cas9 structure-function relationships. PCR-based site-directed mutagenesis is a common method for introducing these mutations. I can provide theoretical guidance on this process, including principles of protein structure, mutation design, and PCR techniques. 2. Designing scaffolds for RNA structure. The design of scaffold RNAs, cRNAs, in systems like CRISPR involves understanding RNA structure and how it interacts with Cas9 and DNA. Mutations in the scaffold RNA can influence the efficiency and specificity of gene editing. I can offer guidance on the principles of RNA structure, the role of scaffold RNAs in CRISPR systems, and considerations for designing mutations. In both cases, the process involves a cycle of designing mutations, synthesizing the modified sequences, either Cas9 or scaffold RNA, and testing their effectiveness in gene editing. I can assist in providing a theoretical framework and understanding of the principles behind these techniques. However, practical design, execution, and efficiency assessment of these mutations require detailed experimental work in a laboratory setting and may involve using specialized software for protein and RNA modeling. My role would primarily be educational, helping students grasp the underlying concepts and strategies involved in these advanced genetic engineering techniques.
Thank you so much.
You're welcome. If you have any more questions in the future or need further assistance, feel free to ask. Good luck with your studies and research.